I really enjoyed that talk. And I think part of that talk was, number one, explaining what FOCUS4 was, and the whole concept of these hypothesis-led biomarker groups, but also talking about the need for this non-stratified chemotherapy arm, because again, Tim very nicely showed that in all of these molecularly stratified trials in colorectal cancer, only a small proportion of patients end up actually going into a biomarker-led arm. So it's really important to think of the design really carefully. Other things he pointed out, he showed very nicely how in over a 10-year period, many things have changed. So how you stratify the patients have changed. So when we started FOCUS4 back in 2011, you were stratifying by, say, just genomic characteristics, so BRAF, KRAS, PIK3CA, whereas actually the molecular field in colorectal cancer has changed over this time. So if you were going to start the same trial now, you'd be looking at different things. So you might be looking at CMS, you might be looking at stroma, and of course the immuno-oncology arm has completely changed. So the landscape has changed quickly, and that's really important for molecularly stratified trials. He also demonstrated the importance of the adaptive and nimble trial design. So you learn from the biology as you go. The trial design allowed us to, which changed throughout the trial cycle in terms of what agents were available to be tested by pharma, and also the biomarkers. He was very explicit about the fact that we did a lot of work, and we had a lot of agents that we were progressing, which didn't actually then make it into the final trial. And this was for a variety of reasons. Number one, the science had moved on. The biomarker prevalence was too small. So when we looked at HER2, it was very small. When we looked at ATM, it was very small. And we had lots of negotiations with pharma, which then at the last minute, the drug dropped down in their priorities. So it was nice to see the progression and all of the ideas that had happened, but actually the very few that had made it into a randomized trial comparison. The other really important point was the endpoint. So progression-free survival was chosen as the endpoint. And the other thing that we were really firm on the FOCUS4 design was the need to randomize a novel agent versus a treatment break, so that you had a comparator. So quite often in these trials, response rate is used as the primary endpoint. So for FOCUS4C, the trial would have been negative if we'd gone for response rate, because after induction chemotherapy, actually the chances of you responding further as per RECIST criteria are quite low, whereas having the progression-free survival endpoint actually allowed us to see activity between the arms. So that was very important. So I think Tim very eloquently showed some of the successes and some of the challenges, which then provides lessons for those of us wanting to move similar trials forward in the future. And we have submitted a manuscript on our learnings from FOCUS4 for publication.